show because I'm running late tonight. I'm here to do a show. And why is my window all messed up? My windows, my tabs.
Yeah, I like these other maps that are out there. They're showing it.
Hello, everyone.
Yeah, Nevada,
Hello, everyone.
come on.
Oh, there are maps out there. Click on the states, click on the maps. Place your bets. No, everyone, hello. This is This Week in Science. At least it should be tonight. It should be all about the science tonight. We know there are other topics on people's minds. It's very hard to not think about. Yeah. So hard to not think about some of these things.
No idea what you're
Yeah,
talking
but
about.
we're going to do a podcast. We're going to do a podcast right now. And in doing this podcast, you know what's going to happen? We're going to talk
You will
about
be
science.
magically transported into a future, a bright one.
A bright and shiny, happy future. Bright and shiny, happy future. Bright, shiny, happy future. La, 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 la. Okay, we're going to do a podcast now, I think. And I'm missing things to say about things. What do we have in the show tonight? Like I said, I am behind on things.
Well, that's a part of where we get to that in a minute.
What?
We get to that part where we say what we're
I have
bringing.
to have three things. Three things.
I hope everyone out there is having a lovely evening.
Yeah.
Really do appreciate you stopping by.
Okay. I can start the show. We can start the show. Okay. We can do this. Okay. I typed things down. We can do this. I'm only running a little behind. Only a little. Am I loud enough? I'm not down far enough. My video looks ragged and I'm quiet. Yeah, I turned myself
you did get ragged
down.
again. Oh no.
I got ragged again?
Yeah.
Look at me, Raggedy Andy. My computer freaked out over something. So why don't you guys hang tight for a minute? I'll be right back. So everyone, this is just a wait on the podcast for a second, but I'll be back with some more volume in just a moment and some less raggediness. Okay.
I'm also trying to fix something. Let's see. For some reason, yeah, I am not in stereo. What's up with that? I don't understand why I am only hearing everything in monotone. Hey Fada, hey Paul Disney, hey Mike Shoemaker. Welcome to the chat room area of This Week in Science pre-show. Be patient, ladies and gentlemen. We are still counting the science stories that we will be bringing tonight. I have 5. Okay, I'm done. What do you got, Blair?
I have
How
3.
many stories did you bring? You have 3? Okay. See, Nevada? That's not that hard. just do the math.
I mean, to be fair, I did start looking for stories yesterday, so I'm in the same boat.
Mike Shoemaker asks, where in the world am I? I'm back in Davis, California, United States. I've been here long enough to have cast a ballot in the election thing that's happening. And it looks like I might not be able to go back to Denmark for a little while.
Oh, because they've started spiking?
They're starting to shut down again. Mm-hmm. Yeah. So I might be hanging out in California for a while. Which is cool.
I would say we should hang out, but we shouldn't.
We should all not hang out together. Wouldn't that be fun?
Yeah. I mean, that's what we're doing right now.
Yeah. Yes, the third world. It very much is. It very much is. In a lot
Oh, yeah. I should silence my cell phone. Oops. I'm a little scattered, boys and girls.
Yo.
You look fine now.
Is it better? Hello. Okay, let me see if I can. I had to restart. I restarted. I went and opened a tab in the Googles earlier, and for whatever reason, opening that tab in the Googles, just at that moment, everything went crazy. Let me get back in my chat room. Are we ready to do this? Are you guys talking to animals? What's happening on the podcast stream?
This is... Oh, are we live? Oh, hello, everybody. Oh, my goodness. Did you just hear all of that? What? You might have some editing to do, Kiki.
No.
Don't worry about it. It's fine. It's fine.
Just
We didn't
let
do
us...
anything bad.
Snippity snips. We're back. Okay.
Told you we shouldn't have played that card game.
I have better video. Sound is better. Let's do a show, shall
do it.
we? Yeah. So let me get my music queued up because that's something that's important. I'm restarting all the computers. Music. There we go. Okay. So let's start the show in a little number called 3, 2. This is TWIS. This Week in Science, episode number 790. Oh, I wrote it down wrong again. I am so... These numbers right now are really not working with me. Numbers. They all count. In 3, 2... This is TWIS. This Week in Science, episode number 798, recorded on Wednesday, November 4th, 2020. Science is all about patience. Hey everyone, I'm Dr. Kiki, and tonight on the show we will fill your head with glowing animals, little nudges, and earwax. But first...
Disclaimer, disclaimer, disclaimer. Math! You either have it on your side or you don't. Whatever numbers you are working with, it's never going to add up to anything other than the sum of its parts. You can wishful think, you can squint your eyes, look sideways at the equation, and if you like, you can stand on your head to get a different perspective. But math has a way of being perniciously indifferent to how you approach the solution. And as ballot counting continues in America, millions await the fate of a nation, now at the mirthless mercy of math. But don't blame the math. After all, without math, we couldn't bring you This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back. Back. Like I said we would be.
Is there a chicken in here? Bark, bark, bark. Bark, bark, bark.
No. Bark. Bark. Cock-a-doodle-doo. Okay. I'm not
What
a chicken. I can talk about science. It's going to happen. We're going to talk about All the science. We are in a mood tonight, and I hope you are in a mood to listen. So, on tonight's show, what did I bring? I've got some earwax, as mentioned in the very start. I've got some missing height as well, and I've also got a few nudges for you. We're going to talk about nudges.
Oh.
Those things that maybe you don't really know are happening. Nudge, nudge. Justin, what did you bring?
I've got early human lady hunters, invasive rats, a leukemia super spreader, ancient Asian genomes, and why magic mushrooms might be a magic bullet in the treatment of depression.
Sounds
Sounded
amazing.
a little too news anchory, actually. But
Very news anchory, maybe a little salesy, but we'll talk about that when it comes up. Blair, Animal Corner, please.
Yes. I have biofluorescent platypus. I have dull-colored birds, and I have surfing fish.
Surfing. If I were a fish, I'd want to surf. Why not?
Yeah.
Okay,
But do you surf on whales? Because that's actually what this is about.
This is fish extreme sports.
Yeah.
No, extreme ichthya sports. All right, let's dig into the show. As we jump on into all these stories, I would like to remind you that if you are not yet subscribed to This Week in Science, you can find us anywhere podcasts are found. Stitcher, Spreaker, Spotify, Pandora, radio.com, TuneIn, all over the place. Apple, Google. You can find us also on Facebook and on YouTube and on Twitch. Look for This Week in Science.
are those all real?
Yes.
Wow.
People have so many options these days. It's
That's
amazing.
wild.
There's also
Just almost any one-syllable word, it turns out, is a web client. So
it is
out there,
is.
especially if there's lots of consonants.
Consonants like calendars. And I want to remind you as well that calendars are available. We'll remind you again later in the show. But yes, Blair's calendars are for sale on the TWIS website. Head to twis.org. Click on that froggy toad. And you can buy a calendar.
Mm-hmm.
Yes, let's plan ahead for another amazing year.
No, just an amazing year. But yes.
An amazing year.
Yeah.
So let's talk very briefly. Let's move into the news. Shoo-choo, short stories. Here we go. Short-ish, kind of. Speaking of missing things, not missing time, but maybe missing explanations for how our genes influence height. Since about the late 2000s, when researchers started looking into the genes of the genome to figure out what markers were linked to various diseases, and starting through twin studies and family studies and looking at the influence of wealth, they started picking out genes that are related to height as opposed to environmental factors like disease state in childhood or how good of food you got or other environmental factors. So the markers that they were looking at, they couldn't tally it up. They couldn't figure out of the DNA markers what explained all the height. They could only at first explain about 5% of its variation. And then they ended up putting together a number more genetic markers, and they got it up to about 40 or 50% of the genetic component of height. So they were getting there. But now, in about 2018, a team of researchers put together an effort called GIANT. It's a global consortium called GIANT, and it pooled the data of 700,000 people looking across 3,300 markers that then explained 25%. They added things up and added things up. But they have been able to put together a bunch of things, and basically they are tracking down up to 80% now of the variation in height based on DNA. It's basically from studies of identical and fraternal twins. And they're starting to put it together. They've identified nearly 10,000 DNA markers now that explain the influence.
And that's why it's so varied. And why
Yes.
you're a mixture of both of your parents, but also it's not a perfect mix. And
why you can't say by just measuring a child and they're measuring the parents that the child will be 6'2" when they are 19 years old. There are a lot of things that go into it. But we've been measuring this for a very long time, and even knowing all of these 10,000 markers that are involved, being able to use them like sliders on a mixing board to produce the height of a child is still beyond our abilities. but we're figuring things out. We're figuring things out. Yes. Yes, so we're getting there. We still have a ways to go. How tall will I be when I grow up? I don't know. I will probably be shorter because I'm already shrinking. Okay, speaking of women, Justin, tell me about lady hunters of the past.
Yes. So men hunt, women gather. When early human groups sought food, the division of labor was distinctly dictated by an individual's gender and how they participated within the group. That's just the way it was, or so somebody must have assumed and then convinced everybody else that became a saying, most likely reflecting the imaginary reality created by men with big sideburns, bushy eyebrows, and extraordinary nose hair mustaches. Because according to the research conducted at the University of California, Davis,
Go Davis!
it was absolutely nonsense. 9,000-year-old female burial in the Andes Mountains in South America reveals her to be a hunter.
Ooh.
Not a gatherer. She's a hunter. Yes. Randy Haas, assistant professor of anthropology, is the lead author of the study, which is titled Female Hunters of the Early Americas, which is published in Science Advances. Quoting Haas here. We believe that these findings are particularly timely in light of contemporary conversations surrounding gendered labor practices and inequality. Labor practices among recent hunter-gatherer societies are highly gendered, which might lead some to believe that sexist inequality in things like pay or rank are somehow natural. But it is now clear that sexual division of labor was fundamentally different, likely more equitable, in our species' deep hunter-gatherer past. So, this comes from one individual that they found, but it was sort of it posed a question. Hey, have we really looked at identifying who in this ancient past was a hunter? So They went back and they looked through all sorts of other individuals, 429 individuals from 107 sites. Of those, 27 were associated with big game hunting tools. So this isn't like hunting squirrels. This is like going after big prey. Of that 27, 15 were male, 11 were not. In fact, or 12 were not, I guess. But 11, or one maybe, there's one missing. I don't know what happened to it. 11 were female. The sample was sufficient to warrant the conclusion that female participation in early big game hunting was likely non-trivial. This 9,000-year-old specimen wasn't just a badass that was participating in the hunting, but it was more of a norm in
more common
ancient times. Yeah. So their statistical analysis shows that somewhere between 30 and 50% of hunters in these populations were huntresses. And even actually in the more recent modern human hunter activities, it can get up to 30%, so it's not completely removed. So anyway, change your views of how you think the division of labor naturally occurs in societies. Because I think like we've talked about this in Neanderthal studies. Mm-hmm. Like everybody goes on the hunt, except for like the wounded that they care for that stay back at the cave or whatever. But everybody goes out and has the broken bones and the injuries associated with attacking the prey.
Whoever's capable, right? It's if you are able-bodied and capable, why not? And I think it would make more sense. I mean, especially in early hunter-gatherer civilizations for anyone who's young and able-bodied to be on the hunter side of things, the gatherers to be the, you're getting a little bit frail, you're complaining about your back and your knees a little bit more. So it's like, Grandma, why don't you stay back at camp, or why don't you go look for the wild radishes?
Now, wait a second. Now, let's not. The gathering required a lot of stooping, a lot of getting up and down, picking things, carrying things along. It was not easy work either.
None of it is easy work. I'm not. None of it. None of it's easy work.
So Thunder Beaver in the chat room is bringing up a really good point too, which is that we know that in Native American cultures, in many of them, there's 3 to 5 genders at least that we've known of. And so that means that their gender roles, if you want to call it that, also kind of ran the gamut in their culture. So you could see how it's all nothing. If that's like really recent too, you can see how it all might be something that's kind of being imposed on what you found.
It's an interpretation of findings, of remains based on current cultural attitudes. And I mean, like the finding of the Viking women warriors that Justin talked about a while back. It's like people hadn't really thought that Viking women were necessarily warriors too. But look at this. Maybe they were warriors, fierce warriors as well.
Yeah. So, but it's also one of those things that you have to remember about, especially when you're talking about a warrior class of the ancient world. Mm-hmm. You didn't need to be like the equivalent of a professional athlete today. You just needed to be well nourished, and you were that much more dominant than most people you ran into.
Right. Yeah, I think yeah, and this just goes into the conversation of modern ideas of gender, and so much of what we think of as just like, okay, we've got man and woman, man works, woman doesn't, woman stays home. This is because of a very it's using women as property, and having it be a dominance through property and ownership attitude, as opposed to egalitarianism, everybody working together and trying to, needing to hunt for the village or the tribe or what, there are many reasons that we have embarked on our current attitudes, which thankfully are shifting. Yeah. Let's talk about the darkness and how we can get away from the darkness. Do I just bring my platypus, Blair?
Yes, in fact, you should. So I don't know if you two remember, but I reported a while back on biofluorescence in squirrels. Do you remember this? The pink squirrels, the pink glowing squirrels. So the same team of researchers that was looking at these flying squirrels and how they glowed in the dark. They were going through all the specimens, the drawers of specimens in kind of the library and the taxidermy library. And while they were doing some of that, they actually decided, hey, let's shoot this UV light on this tray of platypuses that's right next to all these squirrels we're looking at. Whoa, what is that? Are they green? So it turns out they were green, or cyan, which I'm assuming that's a kind of green. Anyway, so they were green under the UV light.
Cyan is like blue.
Blair,
oh, it is?
It's fine. Oh. I'm not colorblind. I have no idea what cyan is.
Okay, great.
I would have guessed it was like a pinkish red rust color. I have no idea.
Well, greenish or bluish, it sounds like. And so they found that these guys were glowing in the dark, which at this point, the only mammals that have been found to biofluoresce are the squirrel that I mentioned and opossums. And now if you're keeping track, we have the 3 different kinds of development in mammals, the 3 different kind of evolutionary radiation, if you want to consider it that way, of mammals. You have the monotremes, which are these first guys that lay eggs, and that's so platypuses, we have some biofluorescence in them. Then we have our opossum, which is our marsupial So they give birth to very underdeveloped offspring, and they go in a pouch. And then we have a squirrel, which is a placental mammal, just like us. So this is something now that has been discovered in 3 mammals, and they happen to be the 3 very different temporally, like developmentally in the evolutionary chain when they broke off, but also developmentally. So now this becomes a question of, first of all, I think we have to shoot UV lights on everything. No,
Everything. Yes.
every
glow?
mammal
Do you glow?
in a museum. Get out your UV light and start taking notes, because either this is extremely widespread and it started in the very first mammals, or it is so helpful that it keeps popping up as convergent evolution, which, there's a whole bunch of questions about why they might want to glow in the dark. The best guess is that they are crepuscular, so they're awake at dawn and dusk. And so are the other animals that have been discovered to biofluoresce, so it might be helpful. But also remember that platypuses hang out in the mud. They move their beak around into the mud. They stir up all this mud into the water column underwater so they can get to bugs and stuff. So maybe it's helpful to be able to see each other through very cloudy water if you glow in the dark. I don't know. But anyway, this also means that they probably can see different wavelengths than we can as well.
Yeah.
So host of questions here. But ultimately, either most mammals can glow, or it just keeps popping up.
Well, I love the idea that they would be more visible during dawn and dusk, where there's a little bit of light peeking in, and maybe that makes others of their kind, they're like, oh, there you are, I can see you a little better. I don't know.
Didn't we, Kiki, maybe you brought it, I didn't, one of you did, that humans slightly bioluminesce?
That there is
ever
work
so slightly, but
yeah. Yep, that we have a glow as well.
We got a little glow to us.
I mean, what if just everything does is the thing? Like, it's a wavelength. It's a spectrum of light. Like, just maybe we just all have some of it.
But then what's interesting is what can see in that wavelength. And what on Earth is seeing this other reality of all the different creatures and how they glow? Is it bees? I don't know what does. I don't know what sees need.
Well, that's also Thunderbeaver again. Just asked in the chat, would you want to see UV light? And 100% yes. I had a black light in my room in middle school and high school because I thought it was so fun.
Of course you did.
Yeah, but also it's more bright, which is closer to probably what the average person sees anyway. It's fine.
It's amazing. It's amazing.
Glowing
one more
platypus.
Yeah, I know. One more thing for this weird animal. Just add it to the bucket that stuff was thrown into at the time when evolution was taking place for the platypus. It was like, shake it all up. Oh, look what we've got here. Very odd monotreme animal.
So fun.
I didn't mean to make fun of you, Blair, for having a black light.
Oh, no, you were in your spot. You probably could have guessed it, and you were right. I had a black light. I had a beaded curtain. I had a lava lamp.
Can I tell you? I had one of those black lights.
I didn't have any of these things. I had none of them.
I had a black light. I put it in my room as a teenager, and I had to get rid of it almost immediately because I also had really bad dandruff.
Oh, no.
And I was like, this is gross. I don't want to see the dandruff on all of everything. Oh, this is terrible.
Yeah, that's awful. What if you wanted to look at your earwax? Do you have lots of earwax?
You check the Q-tip when you're done, right? Just to see
of course.
what
Just
kind
to
of job
see.
you've done.
Yeah. Clean as a whistle.
All right. Well, there is a new methodology where researchers, doctors could easily check the level of your stress by checking your earwax.
Really?
Yes. Researchers from University of College London and King's College in London have published in the journal Helion that about a device that they are using that can be used at home without medical supervision to allow stress checkups and maintain social distancing. It uses cortisol levels in earwax, which the researchers say appear to be stable. That your earwax cortisol levels are there. It's a good, cheap, easy, effective way to get stress readings from a person. Yeah, so normally we can use saliva, we can use hair samples, but hair samples are much more long-term. They're time-consuming, and it's expensive to analyze them. It's cheaper to analyze with earwax. The researcher was actually inspired by honeycomb from bees, and because of that, went looking at earwax, looking to see whether or not it would make a good home sampling source that wouldn't risk a lot of contamination, knowing that beeswax is well preserved and can resist contamination itself. So they have a device that's like a cotton swab, and you stick it in your ear, but it has a break so that you can't stick it in too far and hurt yourself. Good. It's got a tip covered with a sponge, and it also has a solution that is reliable for the samples. So anyway, they found that earwax samples yielded more cortisol than hair samples, and it was the fastest and cheapest method. Earwax for stress determination for the win.
But what if you're so stressed that you're constantly using a Q-tip and then you have no earwax for them to test?
Then you might have an issue.
So this is a reason to not keep clean ears, right? You want to let the earwax build up so that you can test to see if you're stressed.
Let your earwax build. Yeah. Test your stress. Yes, they are working on bringing this device to market so that it could be used as a medical device at home. Yeah, very interesting. Who knew? Cortisol in earwax. What you got next, Justin? Rats? Why do you have rats?
So the Palmyra Atoll, if you're not familiar, is a remote network of some islands out in the Pacific. Once upon a time, it had a rat problem. Now, rats weren't native to this island atoll in the middle of the Pacific Ocean. They likely arrived there as stowaways with the U.S. Navy all the way back in World War II. So they've been 60-ish years. They were there on this island with no natural predators. They basically took over the islands. They ate seabird eggs, the native crabs, whatever seeds and seedlings they could find, and that was just Rat Island from there on out. So back in 2011, the U.S. Fish and Wildlife Service conducted a rat eradication project. They solved the problem. They got rid of the rats. UC Santa Barbara ecologist Hillary Young and her research group had already been tracking at that time another non-native species, the coconut palm. And they were sort of watching the coconut palm as it slowly began to invade the areas. It's considered invasive because it's not from that island, but it's also impacts nesting seabird populations because it changes soil composition, determining what trees can grow there, and the birds, I guess, don't like these coconut palms to hang out in. So they had plots there, and they were monitoring trees in various stages of growth and survival throughout this atoll. This is quotey voice of Anna Miller-Turcule, I'm guessing, graduate student, researcher, and lead author of the study that appears in the journal Biotropica. Prior to the eradication, most of the understory, I guess that's the ground level, of Palmyra was either bare ground, sandy soil, or coral rubble, or covered in carpet of ferns. There wasn't a whole lot of trees. It was sort of barren there. Turns out the rats were quick to eat seeds and young plants coming out of the ground. They frequented the canopy as well, often nesting in the coconut palms and even eating coconuts. So if you can picture this lovely tropical-looking island with palm trees filled with rats.
Yikes.
Really. Okay, so rat eradication 2011. Did kind of work for a lot of things. the Asian tiger mosquito that had shown up on the island left again. It didn't have the rats to feed on. 2 species of land crab emerged that hadn't been there before, I guess, changing the atoll's biodiversity there. The only thing is, the foliage rebounded tremendously in coconut palm. Yeah. Turns out the rats were keeping down the coconut palm invasive species by eating all of their nuts, and so they couldn't grow, eating the little saplings as they would start, and eating the coconuts right out of the trees. They were basically keeping back the other pest.
Huh.
So,
Time to pull out the palm trees.
yeah, that's what they're doing.
Which one do
There's
you get rid
apparently-
of? Yeah, palm
There's
trees.
apparently millions of them now.
Yeah.
It's become sort of a problem. But this is sort of the interesting. This is the happy accident of the experiment is that they got to show an example of when you remove any species, even if it's an invasive species, if it's established, there are downstream effects that shift the ecology in other directions that you may not be anticipating. So.
I think that is the key point also, is the fact that biologists like to think they know the ecology of a habitat, of an area, all the things that are in it, but there are usually, not always, but usually unforeseen consequences. The ways that that organisms interact with each other. It's it's like the genes and the markers in our genome related to health. There are over 10,000 markers related to height. There are over 10,000 markers related to height. How they interplay and how they interact with themselves and with other things in the genome. It's the same kind of thing. The ecosystems are complex.
Well, and it's just like any problem, you can't almost ever boil it down to one thing, right?
Exactly.
It's always there's always a multi-layered way to solve any problem.
Yeah.
And there is a tendency to tackle these problems as a one species problem. And, it's the Fish and Wildlife and also an island or nature conservatory, island conservatory groups that were involved in the eradication that identified the rat as a problem and what they can improve. They weren't obviously tracking the foliage, the plant, So even within them, they saw no other species that this was going to conflict with in the animal kingdom. And it looks like they're right. The mosquitoes weren't native. They're gone. These other crabs move in. All of that work, they're like, aha. But, it's slightly outside of your discipline. Just one adjacent. You got to look there, too.
Yeah. It reminds me of our conversation a couple of years back with Jess Phoenix, how she was talking about her project Blueprint Earth and how they're working on trying to get a blueprint for all of the different of ecosystems. What are all the things that are there? So it's not just the wildlife biologists talking about the wildlife by themselves and not talking about the plants with the plant biologists, and then you have the crab biologists.
Well, and that's the thing. It's like if this had been anticipated, if this could have gotten put together and anticipated, you could have pulled out many thousands of trees before you got rid of the rats.
Mm-hmm. Yeah.
Instead of now millions and having the thing really. So it's. Knowing that the things are going to walk hand in hand allows you to be more proactive and use your resources better if you're trying to do a conservation effort.
Yeah. Something that has been conserving its batteries, has a big conservation effort, is Voyager 2. The Voyager 2 spacecraft, which has been traveling away from Earth for more than 43 years, is now 125 astronomical units away from Earth. Astronomical unit is the distance from the Earth to the sun. It's 125 times the distance that we are from the sun right now. It is outside of our solar system. It's moving out of our solar system into interstellar space. It's out there. It's been doing that. And it was quiet for a very long time. It's very quiet. And it just called home. Yes. So last week, to test new hardware that was recently installed in the Deep Space Network, Voyager mission managers were able to send a series of signals to the spacecraft for the first time since March. And Voyager 2 called home and said, yeah, Mom, yeah, Dad, I got your message. I'm out here. I'm just fine. Doing my year abroad. Getting out there. Yeah. So Voyager 2 is still doing well, venturing out there beyond the solar system, investigating the great beyond. And it's just amazing that we have Voyager 1, Voyager 2, and now the New Horizons mission that are really out there, going out into the outer reaches of the solar system, beyond our solar system, and exploring. There's some new stories this week about thousands of Earth-like planets around, or at least potentially habitable planets around Earth, around stars like our own. And so if we could only get there, but we can imagine. And we can send our robots.
So Voyager's going out there. We just found out we can hear back. So that means that we can now grab some data from...
Yes. And we have been getting data from it on occasion, but because they were updating things and doing some new dishes and software and stuff, it's been a while since March that we've heard from Voyager 2. And, in the times when Voyager is offline and not responding to our signals, it's kind of, it's just recording stuff.
Like
it's got its batteries, its little nuclear power that it's... that it's moving on, and it's heading out there.
So Voyager 2 is gonna keep sending us stuff, but at some point it'll be too far away.
Maybe. Well, I mean, it'll just take a long time for information to get back. Or there may actually you're right. At a certain point there is decay. If the signal is not a strong enough signal to begin with, then you have signal decay over time and space, but it's going to be a long time.
I'm not reading about how many astronomical units, because I think it's 90 million miles to the sun, something like that. I mean, that's 10 billion miles away. It seems like it's
got some range on it.
Yeah.
Yeah. Hopefully we can
get
it out.
That's back when we had a thing called Radio Shack, and you could get NASA went down and just got these little signal boosters. It used to be a thing. It was really easy.
I mean, that's the technology that was used in the 60s and 70s. We think of it as very basic, but compared to modern technology and the strength of the computers in our handheld phones. But what's really amazing to me is that because of the small and delicate nature of technology today, it's more destructible. So interstellar or solar radiation can damage the technology that we use in the transistors in the silicon chips that we use today. So you have to use more shielding. They have to be more protected. And those old spacecraft that we sent up, they were Radio Shack. It's put them together, let's get them out there, and it's going to keep going. It's just solid parts. They're going to work. Just like this show, which just keeps going and going, episode after episode, year after year. Oh, hey, if you want to help us keep going, head over to TWIS.org and click on the Zazzle link. I mean, buying stuff in our Zazzle store will allow you to get some cool stuff, but it's also going to support our show and will help us keep going and do the things that we do. Also, while you're there at TWIS, don't forget we have our calendars for sale. Dun, dun, dun. Blair did a good one. Yeah. Okay, should we talk about COVID now? Is it time? Let's have that.
Please, something relaxing to talk about. Finally.
Can we? Because I don't know if I've been talking about anything but COVID for the last 8 months. Dear, oh dear. All right. There was some good news several months back about antibodies in llamas. Did you hear these stories about llamas having antibodies that could neutralize the SARS-CoV-2 virus? Perfect little antibodies. Well, they have these, llamas do. They have these little antibodies that are called single domain nanobodies. Single domain antibodies, otherwise known as nanobodies. Nanobodies are found in camels and llamas, and apparently they had some good ones against SARS-CoV-2. Great. Well, we can't go around harvesting these nanobodies from llamas because, number one, there aren't enough llamas and camels on the world. It's expensive. It's difficult to raise all the animals for all of the antibody that would be needed to neutralize infections in the entire global population.
What if I can get my hands on a llama?
You got to get your hands on a llama.
How much is that worth if I can get my hands on one?
Oh, everybody, if you've got a llama, lick your llama today. I mean, like your llama. Never mind. Don't listen to me. But listen to me about this science. So researchers have been trying to figure out how to get the antibodies, the nanobodies, without using llamas, without using camels. And so they've been searching for synthetic versions of these nanobodies, which are called Psi-bodies. Nanobodies to Psi-bodies. And Psi-bodies could be potential alternatives. Well, a group of researchers at EMBL Hamburg have used a platform of Psi-bodies to find some Psi-bodies that work to neutralize the SARS-CoV-2 spike protein. And so they think that this, they had six of their analyzed Psi bodies, SB12, SB23, SB42, SB76, SB95, and SB100, bound the spike protein with fairly high affinities, and these affinities are pretty good. So the Psi bodies would be less expensive, easier to create than getting them from llamas. And the scientists performed a neutralization assay using a bunch of particles that were modified to carry the spike protein on the surface. And they say that 36 Psi bodies were screened for neutralization, identifying 11 that are capable of neutralizing SARS-CoV-2 at a fairly high rate. So they found one in particular, SB23. It's a very potent neutralizer, and we'll see where this goes. Not the llama bodies. It's a Psi body. All right. Beyond Psi bodies. Oh, poor minks. Justin, you made it. You made it out of Denmark, but the minks aren't going to.
No, but minks.
Poor minks. We've talked before about the other animals that have a high probability of succumbing to COVID-19 infection. Humans, cats, minks are among them and can catch COVID-19 at a very high rate. And in Denmark now, over 1 million mink have been culled in the country as a concern for the mink, and also that the mink will transmit the virus back to humans and could potentially be a sink where mutations in the virus could take place.
Which
Big
I
concern.
believe has already happened, hasn't it? I mean, I believe that there was an outbreak centered around mink farmers.
Yes, there has been.
Okay.
Yes.
So yeah, it goes into the mink. Then there's the other mutation. Then when it gets back into the humans, you might be dealing with a slightly different SARS-CoV than you had to begin with.
Right.
That's not cool.
I mean, maybe it would be a less transmissible version. I don't know.
That would be great, but
less bad for you version. The mutations
another
go
version.
all directions.
Do you want to watch that movie again? I don't.
No, let's not do this again.
No.
Yeah, but Denmark is among the countries that has definitely put a call from the government down on the mink farms to kill their animals because of concerns for infection, which is bad news for minks. But tell me about this leukemia super spreader, Justin. What's going on there?
Okay. So this is a case study that details a leukemia patient who was actively shedding COVID for 70 days. So
It's a really long time to be actively shedding the virus.
Yeah, it's a really interesting. It's sort of a really interesting case. Not completely unique, but very interesting. This is in the journal Cell. It's an unusual case of a woman with leukemia and a very low antibody count who was infected with coronavirus for 105 days at least, and was actively shedding that virus for at least 70 of those days, all the while remaining asymptomatic the entire time, despite being 71 years old, something along that lines. So this woman is, I believe, from Washington, and was in a nursing home that was heavily hit. One of those first, one of the first things we heard about in COVID was nursing homes in the Seattle area. She's one of those. So she had gone to the hospital despite having no symptoms from that for some other reason. It was anemia, which is another thing that she had. So she has immunocompromised with chronic lymphatic leukemia. She goes to the hospital. They go ahead and test her because of where she was. Sure enough, she's got the virus. And so the first physician attending wanted to know if she was still shedding this virus, if it was still. So they got other people involved. Sure enough, she has. Munster, who's one of the authors of this study and one of the ones who's been tracking her, Munster says that as far as he knows, this is the longest case of anyone being actively infected with SARS-CoV-2 while remaining asymptomatic. We've seen similar cases with influenza and Middle East respiratory syndrome, which is also caused by a coronavirus. We expect to see more reports like ours coming out in the future. And she was getting the PCR tests throughout this entire period. So it was, apparently they think maybe because she's immunocompromised, her body never made any antibodies, but it also didn't have the
inflammatory response. It didn't have the inflammation that is the problem.
Exactly. It's
that
secondary.
Like, I can't. I've had enough.
Yeah.
Yeah, resistance is futile. Yeah, I won't resist. Oh, well, we'll keep looking.
You just get in there. That's fine.
I'm just maxed out. I can't take another thing. I'm sorry.
But see, now, when we talk about testing, here's a fantastic example of why absolutely you'd want to test. Because there could be asymptomatic, meaning not coughing, not sneezing, no fever, none of the issues that have been reported as associated with the illness, but actively shedding for 2.5 months. How many people can there be who fall into this category, who are super spreading everywhere they go, but would never know it because they never got sick?
Right. Yeah. Testing would be so important. And there are probably many more who have compromised immune systems in one way or another. And
Well, and this is also why, right, the even though they say if you want to be safe, you quarantine for 2 weeks after you might have been exposed or whatever. But when we talk about if you did a real shutdown, it would be more like 6 weeks, is because you have to account for the fact that people spread longer than 2 weeks in outliers.
Yeah. Final COVID story for this week. Results published in the Lancet's eBiomedicine journal from the study of the lungs of 41 people who died from COVID-19 in Italy found that their lungs were messed up. There was the normal lung structure was distorted. There was scarring of tissue, and there was a lot of blood clotting in the arteries and the veins. And the blood clotting of all of these together, but the blood clots especially, suggest a reason as to why the people we call long haulers take so long to recover from fatigue, from shortness of breath, and end up at a place where it takes months for the lungs to recover and the blood clots to clear for normal lung function to happen again. So this was a study out of the University of Trieste and King's College London, looking at these lungs from February to April. All 41 had extensive lung damage. 36 of the 41 had massive abnormal clotting in the lung arteries and veins. And 90%-ish of the lungs had a fusion of smaller cells into giant single cells with lots of nuclei, like the cells fused together. This is caused by COVID-19's spike protein, this fusion into what's called a syncytia. And
I didn't know that was a thing. Wow.
Yeah, it changes the cell membrane structure, stimulates the fusion of these cells with infected cells with normal lung cells, and that can lead to the structural changes. There's then inflammation and this blood clotting that we've been talking about. So this is a really interesting study, actually digging into kind of what is happening in the lungs and potentially pointing to an explanation for long haul effects, which we still really don't understand yet. But one step at a time. It's not pretty. And you know what? It hasn't magically disappeared yet, and we'll keep talking about it as long as it's here and as long as we're learning about it. Just letting you know it's a reality check right now, everybody. Additionally, there are many of our listeners are in the UK, and I know that the UK is potentially going into a lockdown because of their COVID situation at the moment. So I would love to know how you and our audience feel about lockdowns. What do you think? I'd love to hear from you and to get an idea about the variety of thoughts that are out there about lockdowns. So if you could write to me or send a TWIS message, or should I do a Twitter poll? Maybe I'll do a Twitter poll.
Yeah.
I'll do a Twitter poll in the next 5 minutes. And if you can all respond to my Twitter poll, it'll be out there. And I'd love to hear from you. And if you're in the chat room right now, you can let us know how you feel about lockdown and whether or not you think it's a good idea. Because we have talked about it a lot. Yes. All right. Hey, this is This Week in Science. And if you want to help us grow, maybe, just maybe, you could tell a friend about us today. That would be really cool. Share us with your friends. Just like your favorite pair of socks. No, that's not. Nope, that's not right.
Wait,
just
what?
Like
no, I'm not. My favorite socks. No, maybe my second favorite or third favorite I might consider, but not my very favorite.
No, share us with your friends just like a 6-pack. There's enough TWIS to go around.
From 6 feet apart, though.
From 6 feet apart, yeah. Gosh darn it, I'm just not going. Okay. Do you know what time it is?
What
is that time again?
It's a certain time of the day when we like to sing a song and talk about animals. It's Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
So we talked earlier about brightly colored biofluorescing animals. I want to talk about drab, dull-colored animals. We're talking about birds.
Because those are the ones that you can see. No,
yeah,
wait.
I mean, I see it the same way you see it, which is nice.
Wait a second.
When we think about
birds.
Yeah, when you think about birds, you're usually thinking about like parrots and birds of paradise and zebra finches and all sorts of fun colored birds. But there are some birds that are kind of drab looking, just kind of dull colored. And it turns out that the way that they see colors might be different too. So this is out of Duke University, and they tested the ability of different birds to categorize colors and found that it is not universal across birds. They selected 8 colors varying continuously from orange to red. So this is already something I would have failed. And they made little discs with either one solid color or a combination of 2 colors. Birds were then trained to flip over the discs to show the 2-colored side in exchange for food. So no matter which side it landed on, kind of randomly, they have to change it so it has the half and half showing. But the idea is the closer those 2 hues get to each other, the harder it is to tell which side is solid and which side has the 2 colors. So this is how the researchers were able to look at what is called categorical perception. So there's a continuous variation, but we bin it into separate categories. So I immediately started thinking about when I do the calendar, for example, I have colored pencils that I use this year, and I have a 80-piece set of colored pencils, and I arrange them in Roy G. Biv, basically in a rainbow. But if you look at them, you would still call it purple, blue, and I'm just naming colors in the wrong order now because that's how bad I am at colors. There's a red section, there's an orange section, there's a yellow section, right? But as you get to those borders between the 2, like between orange and yellow, there's some that are not quite orange, not quite yellow, right? But you still, in your brain, if you saw that separately, you would call that orange or yellow. You'd categorize it. And so in this study, the female zebra finches, who they started with, presented with a continuum of orange to red colors. They categorized them in 2 distinct groups. Basically, they saw red, this is red, and this is orange. And this would track with their ability to see something that is relevant to them because they have orange and red beaks. And so that would kind of correlate to their need in the wild. So this is a signal of health where females are choosing their mate, how brightly colored their beak is. So then they looked at Bengalese finches. They are brown, black, and white, and they don't rely on colors when choosing a mate. They actually rely on song when they're choosing a mate. So when they were presented with the same continuum of colors, they showed no evidence of a threshold between orange and red. This side is this stuff is orange over here, this section, and that section over there is red. Instead, they only picked discs that were on opposite sides of the continuum and totally different in hue. So basically they were me, right?
Yeah, they were colorblind.
Yeah. So they
funny.
paid more attention to the differences in brightness than the zebra finches. So they were looking at brightness, but in terms of the zebra finches, they were just looking at brightness as a way to reinforce differences in hue for the zebra finches.
Right.
So the way that these 2 different birds see the way that they see color seems to be very different, or at least how they categorize color in their brain. So as far as they can tell, their eyes and their brains physiologically are not any different. So it has to be about what's going on internally in their brain, right? Exactly. Yeah. So it has to be somehow behavior related. So one of the things that they suggest is that Bengalese finches and zebra finches are both domesticated, but Bengalese finches are more distant from wild finches in terms of how they were bred to look the way that they look. And so it's possible that in the domestication process, they have kind of lost the ability to perceive because it became less and less relevant. So there's one theory that it has to do with domestication, but the other theory is just that it's the whole nature versus nurture, it's both thing. So they have a lot of the same equipment to be able to see colors, but it's not relevant to them as they grow up. So they brought this story because I actually think it's really relevant to what happened to me when I got my color blindness sunglasses. So the way it works is when you get them, you have to wear them for hours the first time you wear them before colors get to their full intensity. And it's because you're using part of your brain you've never used before.
Right.
It's not your eyes that need adjusting. It's your
brain.
So the sunglasses fixed the physical problem. And then your brain has to adjust, has to like gain plasticity to acknowledge new data that it's never had before. And so I think this seemed very similar to that. They might have the ability to see different hues, but it's unnecessary for them. And so they've never really put that much effort or thought into it, right? Of course, I'm anthropomorphizing now, but basically it was not part of evolutionary success to be able to tell hues of color. So they don't have as much, I don't know, mental space, mental energy, practice, any of these things that help them to categorize colors effectively. So that's kind of the giant leap from this small study of 2 species of birds.
Right.
So again, this is a lot of speculation based on preliminary study. There's a lot more that needs to happen. They need to see if they can see something similar in wild birds, or if they can expand it to more species of domesticated birds. We know there's a lot of finches out there, so you can just start pulling finch species and seeing how they can do with colors, and if they have color signals in their mating process. And you could kind of try to draw these correlations and go from there.
Regardless of the mating aspect, there's also the side of it that food is colored. It's not drab, and being able to see color and recognize color and respond to color in the environment is that's an important thing to be able to do. So I think
when that's where this domestication thing could come in.
Yeah.
So maybe it's compound,
there's little
right?
Colored beads and seeds in that store-bought bird food that you feed to finches, the finch mix.
But I guess evolutionarily speaking, are you going to die if you can't tell them apart? No. It's all edible.
Yep.
There's no predators. You just get fed and you shovel it into your mouth. That's it. So it seems like evolutionarily, it might not matter to be able to see colors if you're domestic species, right? But so again, this would be a good reason to look at domestics versus wild birds and see if you see similar things. But the headline here is that the more dull colored species was not as good at telling colors apart, which it seems like a pretty funny, weird, far out headline, but the implications when you dig down into it are pretty interesting.
They are very
Are they better at
interesting.
seeing muted variations in color? That would be that's what I would want to know. Are they just?
Yeah, that's a good question.
If they did the same,
if they did the same thing or if they did the same thing but with different types of patterns, right? Like, are they better at catching up on other visual cues? Yeah, great question.
I find it very interesting because then there's like the flamboyant cuttlefish with all those colors and its amazing eyes. So, the flamboyant cuttlefish is like, let me see everything. I have multiple color receptors and
Right. But then if given enough time or domestication processes or whatever.
Would the flamboyant cuttlefish become drab cuttlefish?
Yeah. Or would they stop recognizing flamboyance? Heaven forbid.
Never.
Well, speaking of flamboyance, how about a fish surfing on a whale underwater?
What?
Yes, tell me more.
Yes. So this is a study from a team of an international team of researchers. They set out to study the fluid environments of blue whales. So originally they wanted to see, they wanted to do fluid dynamic analysis of whales. They could see what could be used as a basis to better understand behavior, energy use, ecological health of the species, and ways to improve tagging and tracking of whales and other migratory animals for future studies. But what instead they found was surfing underwater fish. So this is from underwater fish as opposed to above water fish. Oh boy.
Right, like, yeah.
Remora fish. They're also called sucker fish. It's because they have a sucker powered suction disc on their head. And they are known to stick to the body of sharks or other large marine life like whales. And in the researchers studying the fluid environments of blue whales, this is off the coast of San Diego, they were able to capture the first ever continuous recording of remora behavior on a host organism. And they did that with biosensing tags with video recording capabilities. So they were able to have these kind of long-term data points of not just visuals on these surfing fish, but also the fluid dynamic stuff that I talked about before comes into play. Because how, as a fish, do you hang on to a whale as it's swimming, which is pretty fast,
Very carefully.
and very carefully, and they move around while it's happening? So kind of like the surfer who's switching around his position, his or her position, and like going up on their hands and stuff. Well, in this case, the remoras move around, they feed, they socialize, they move all around that whale as it's swimming. How the heck are they doing that without getting flown off into the ocean? Well, they have some really intense fluid dynamics working in their favor as they are sucked onto the whale. So the whales sometimes go more than 5 meters per second, which is pretty fast. And so it all comes down to the drag on the whale. First of all, they select the best spots on the whale, which if you're a remora, the best spots to hang 10 or hang fin,
20,
I guess.
fin,
I don't know.
right.
It, right behind the blowhole. And then also behind the pectoral fin and behind the dorsal fin. And so these are the areas that I guess because there's, the fins make sense because they're reducing drag right in front. So
Yep.
they're catching all the water that's moving. And then the blowhole, there's in and out move, or there's out movement. So I guess that's also going to create. Plus, I think there's a little cusp on the blowhole. So that's all going to mess with the fluid dynamics around that area. So it reduces drag. So the drag is actually pretty low. So if they hover off of the whale for a second, they're not just going to get pulled away in the current. But they also can move around to those different positions out on the whale. So you can see, those of you watching the video that Kiki's playing right now, they can move all over that whale at about 1 centimeter above the whale's skin. And so the way they do that is, I'm going to say a whole bunch of sciencey things now. Bear with me.
Go science.
Okay. In this narrow space between the remora and the whale, when fluid is funneled into a narrow space, it moves at higher velocity but has lower pressure. So it is not going to push the remora away. It actually sucks it back towards the host. So they can swim up into the free stream to grab a bite of food and then come right back down to the boundary layer. And it takes more energy to swim into the free stream flow than it does to just move around where the whale is. This is called the Venturi effect.
Yeah. This is
Fluid dynamics.
fluid dynamics used by boats and planes and all sorts of things. Yes, this is, and now we know by remoras, and it's basically once the remora is a passenger, it stays a passenger because that's just the best place to be.
Absolutely. And so now researchers can take this knowledge and use it to adjust how they tag whales, because when they do tag whales and other large aquatic animals, they often have all sorts of problems. Either they're too bulky, they might impact their behavior, they fall off in a couple of days, and that's a huge amount of money and time and effort to catch that animal, to tag it, and then it's gone after 2 days. So if they can use this information to make a new kind of tag, that would be a great way to collect long-term data. And also it just, it answers a lot of questions about the remoras. they've only ever had pictures, still images, and anecdotal evidence of this behavior. So this is the first time it's been caught on tape.
Kevin Jones in the chat room asking for a friend. What happens to them when whales breach? Which, yeah, like that might be. Question. That might be, especially they breach, but then they kind of like slap the water pretty hard when they come down. Maybe that's the whale's way of saying too many.
So I would wager a guess that the remora can probably feel the upward movement and would scoot over to the pectoral fin. So that a lot of time the pectoral fin does not breach. So they might be able to move their position on the whale so as to stay on when they breach. I don't know. It's when they're going fully out.
This question.
I don't know. They might not make it.
I don't know, but if you're just hanging on and you're a passenger, why not? I mean, fish can be a fish out of water.
Are they doing a symbiotic thing? Are they like cleaning the whale
I
barnacles or something?
I bet the whales don't even know they're there. Although, actually, to answer that question about the cling, I do have a bit of actual numbers about that. So, the force of a remora's suction is about 11 to 17 newtons, and the force that a remora experiences when they are in the most intense place on the whale, underwater again, is 0.14 newtons. So they're hanging on pretty tight.
This, engineering overkill. That's the way you do it.
Yeah. Well, behind the blowhole, where they hang out the most, it's only 0.02 newtons. So yeah, definitely unnecessary. But maybe that's why. Maybe it's for the breach.
No, over-engineering. Oh, yeah, maybe all about the breach.
Yeah.
Let's go into the breach with Patreon. If you would like to support This Week in Science, you can help us keep doing what we do by heading over to twis.org and clicking on that Patreon link and choosing your level of support. With your help, we can keep bringing a sane perspective, a sciencey perspective to the world that is kind of full of misinformation and disinformation right now. So be a part of helping us to combat that and bring science to the world. Thank you for your support on Patreon. Thank you for your support of TWIS. We really can't do this without you. We can't. Coming on back right now, it is time for Justin. What stories do you have?
So this story was sent in to TWIS by Minion Dave, who reminds everyone, time flies like an arrow, fruit flies like a banana. This is from the Max Planck Institute for Evolutionary Anthropology. Researchers have analyzed the genome of the oldest human fossil ever found in Mongolia, 34,000-year-old woman, who the DNA analysis show inherited about 25% of her DNA from Western Eurasians, demonstrating that once upon a time people could travel internationally. Ancient DNA extracted from the skull cap shows that it belonged to a female human who lived 34,000 years ago and was more related to Asians than Europeans, but had that connection still. Comparisons to the only other really early East Asian individual who was genetically studied to date as a 40,000-year-old individual who was found in a cave outside of Beijing, China, shows that those two individuals are related to each other, although the Beijing fossil does not have the Western Eurasian relationship in its ancestry. Interestingly, both individuals carry DNA from Denisovans, the extinct hominids that inhabited Asia before modern humans arrived there. Quotey voice, this is direct evidence that modern human communities in East Asia were already quite cosmopolitan earlier than 34,000 years ago, says Diendo Massolini, lead author of the study and a researcher at the Max Planck Institute for Evolutionary Anthropology. This rare specimen shows that migration and interactions among populations across Eurasia happened frequently already some 35,000 years ago. Researchers used a new method developed by the Max Planck Institute for Evolutionary Anthropology to find segments of DNA from extinct hominins and other genomes. They found the two genomes not only contain Denisovan DNA, but also DNA from Neanderthal, which is also interesting because Denisovan and Neanderthal are sort of themselves ancient cousins that split off long before current modern humans got to the area. But it also shows a path, perhaps, of out of Africa, mingling with Neanderthal, then keep going, mingle with Denisovan, keep going. So anyway, research is reported in the journal Science, and it is just continuing evidence that regardless of where you are on the planet, humans are a braided stream of evolution that isn't just the current modern human evolution braidedness either, but includes all sorts of archaic and other ancient human offshoots that cycle back into the genome that makes us us.
I like to be us. I'm glad there were lots of others before us to make us us. That's a cool find. Very, very cool.
I'm acting as though I'm thinking heavily. I'm going into my next story, but it's taking a moment to load. According to the National Institute of Mental Health, there we go.
Good move.
According to the National Institute of Mental Health, more than 17 million people in the United States and 300 million people worldwide have experienced major depression. The rest of humans are just shallow and unthinking people. They just don't have feelings. Small study. This is a very small study of adults with major depression. Johns Hopkins medicine researchers report that 2 doses of the psychedelic substance psilocybin given with supportive psychotherapy produced rapid and large reductions in depressive symptoms. Most participants showing improvement, and half of study participants achieving remission through a 4-week follow-up. So remission in something like this is more than you expect from any form of therapy or treatment or medicine that is on the market, so to speak. Compound, which is found in so-called magic mushrooms. Psilocybin can produce visual and auditory hallucinations, sometimes profound changes in consciousness and perspective over just a few hours of ingestion. Back in 2016, Johns Hopkins medicine researchers first reported that a treatment with psilocybin under psychologically supported conditions relieved anxiety and depression in people with life-threatening cancer diagnoses. Now the findings from a new study, this is published in JAMA Psychiatry, suggest that psilocybin may be effective in a much wider population of patients who suffer from major depression than previously appreciated, although it's just extrapolating from, cancer patient psychology shouldn't be that different than general public psychology when it comes to anxiety and depression. Small group, only 24 individuals. 67% showed a more than 50% reduction in their depression symptoms at the 1-week follow-up. 71% showed the reduction at the 4-week follow-up. Overall, 4 weeks post-treatment, 54% of patients were considered in remission, meaning they were no longer qualified to be called depressed.
That's amazing.
Really incredible. So
amazing.
This is one of those things that was initially used, and we've talked about this in the past. Initially used in a psychiatric environment with amazing effects. Then fell into the hands of counterculture and ran up against a government that was afraid of counterculture, and so was made illegal. It was technically just legalized in the state of Oregon.
In Oregon, where I'm located. Yeah, it's
as well as for research. It's been opened up there for research purposes.
Yeah, it's for medical treatment, so research purposes and for for medical treatment for conditions like depression and anxiety. So
I think
it's awesome.
also
It's not
mushrooms,
for recreational use.
Yeah, I think also mushrooms have been decriminalized in Washington, D.C., which...
In Oregon, we also decriminalized, I think, all
Everything.
recreational drugs
Everything.
this election. Yeah.
I think it's interesting that the way medicine, pharmaceuticals, I guess I should say, has kind of moved in the modern era is like you have all these natural remedies that have been used historically by native cultures and all sorts of people, and then there was a push to distill down exactly what chemical it was or find a chemical compound and make it synthetically. So there's this huge kind of swing that way. And now we're developing kind of a little bit of a swing back where we're looking at the natural origins of things, and maybe that's not so bad, which I think is great. It's as a pendulum always works, we should really be in the middle. We should be looking at like the scientific implications of a thing, and then, you know, maybe not trying to mess with it too much, because now we know, like, maybe you find what you think is the thing in the mushroom that is the beneficial thing, but if you make it in a lab, maybe it won't work the same way as the mushroom did. So there's, I don't know. I think that
I'm a lot more cynical.
Layered.
I'm a lot more cynical. You know, my understanding is the part of why cannabis was removed from being used as medicine and pain relief was because there were pharmaceutical companies that know couldn't patent it, but did have a patent on something else they wanted to sell.
That's where that's
this one didn't really kind of run up against that. This one got made illegal while research was showing how useful it was for a completely sort of societal, political kind of a reason. But then we leap forward into, what would it be, the late 80s, early 90s, when we have all of these psychotropic drugs, pharmaceutical drugs that are meant to deal with anxiety and depression, some of which can create, led to suicides.
Other problems.
Tons of other problems.
Addiction, all sorts of problems.
I don't remember so much of the addiction ones, but I remember there was a lot of mental health side effects that came with it. I mean, I remember tremendously horrible stories about teenagers committing suicide while on antidepressants.
Right.
Because they had these horrible side effects. This is one that hasn't been known to have those side effects. There was another one that also said that it increased empathy that seemed to last for many, many years after somebody used this, like having empathy.
Yeah.
Like, should we put this in the water? But but yeah, this has always been, we've talked about this, I don't know, maybe a dozen times over the years. This is always one of those available treatments that has been clinically shown to have a tremendously positive effect and has been illegal for the recreational aspect. And why must there be that sort of line there at all? Why can't you still make it illegal?
Fun is bad. Recreation with your brain is bad, because then you're not producing with your brain. Producing is good.
Something forbid that we all think for ourselves. But
that's
amazing.
This is moving tangentially away from the science. But yeah,
it's very exciting.
It's
that
exciting.
The law has changed. The law has changed. No, it's not. The law has changed in Oregon, so the science is actually going to be able to be pursued after a 60-year hiatus from reason.
Hopefully it leads to research happening here in Oregon. Hopefully federal law does not come in and negatively impact this state decision. But yeah, we'll see. We'll see where it goes. Things that can help people are always a good thing. Let's talk about helping people, changing people's minds. Do you know how much corporations change our minds? So it's ridiculous. It's really, really ridiculous how much corporations are changing our minds, influencing our decisions, sometimes without us even knowing it. A researcher published a new study this week in the journal New Media and Society. Researcher Dr. Tanasia is a professor of media at Illinois, and the co-author Wu is a professor of media culture and communication at NYU. Their study is called Going with the Flow, Nudging Attention Online. And in the title, flow that they reference is a concept that used to be applied to radio and television, the audience flow. And that was something that was defined by programmers who programmed the content for television and radio. Nowadays, we've got a la carte stuff, and you can hop from Netflix to Amazon Prime to, Hulu to YouTube to, wherever to get your content. But there used to be a very directed flow that would take people through the evening and keep people on their channels and watching their shows and seeing their advertisements and hopefully buying things and having certain perceptions and perspectives as a result of seeing what they watched there. And so they wanted to take that and apply it to the internet and to see whether or not there are predictable patterns of how people go from website to website and how that happens. And so they took a bunch of data from a data research company, ComScore, back in October 2015. And their sample was based on 1 million internet users, including 1,761 websites that reached at least 1% of the U.S. users during that month. And then because of all this data, they were able to identify clusters or constellations of websites that represented sequences that were common within behavioral patterns for users, for browsers. And it was all browsing behavior that was linked to corporate ownerships, partnerships, and website types. The researchers say that they identified 11 clusters and the anchor sites within these clusters that served as the common starting points for how people start their browsing and get going. And among the clusters are Bing Microsoft, which is anchored by Bing and MSN content sites. A Google cluster, of course, anchored by Google Search. A lot of us use that, and the Chrome browser, YouTube, and Gmail, and a social media cluster that's anchored by Facebook, Twitter, and LinkedIn. And then there were two other clusters, the Yahoo cluster and the AOL cluster, that they surprised the researchers because nobody thought they were going to still be relevant, but they think it's possibly because of older users using these clusters. There were...
Hey!
I
am part of the Yahoo cluster. I admit it.
Do you search with Yahoo?
No, I don't. Oh, okay. I don't search with it. No, I Google everything. I only Google. I don't even like results. When I get results from other browsers, I'm like, that's not how Google would give it to me.
That's not
Get
how
out of
I like
my
it
way.
like that. Go away. There were some other clusters too. They were surrounding data solicitations, retailers that use Citibank as their banking backend, pornography sites, job search, and travel. These were various browsing clusters. And there were 4 methods that corporations nudged online users. And this is the word that they use throughout the study, is this nudging, that corporations are using technology, using the websites, they're using wording, they're using all back-end stuff to nudge people in certain directions online, and we're not always aware of all of this. Then these methods, they're at a different level of visibility and control. And so the highest was content ranking and curation. And so search engines and social media do that. You do the Google search, what's at the top of the algorithm. The search engines are basically ranking things and telling you where to go. Social media also have an algorithm that tells you what to look at based on what they think you should look at. Then there's hypertext. These are words that are linked within the text. And additionally, the nudge is visible, but I guess the users don't have as much control. Third type of nudge is employed by Microsoft through software configurations that are built into the Windows operating system itself, like making Bing the company browser, search engine, and homepage, all by default. And that when it's in there, people aren't necessarily going to change it, right? Whatever's on your computer, you're like, oh, just use what's here. And so that nudges people into certain browsing patterns. And then finally is the back-end databases and software. That's e-commerce, like Citibank, and there's credit card payment retailers. And then additionally, there's all sorts of stuff that's on the marketing side of things where there are funnels, and people talk a lot about funnels that you get pushed down if you click certain links moving certain directions. So really it's that people don't see the internet as a space that operates in this kind of behavior directing way. People think they're directing their own behavior on the web, but there are actually lots and lots of ways in which we are being nudged by the corporations in the world that own the pipelines, that own the web spaces to do what they want us to do.
Mm-hmm.
Welcome to the machine. What did you dream?
Welcome
It's all right. We told you what to dream.
The machine. Yeah. Welcome to the machine, everybody. Oh, yeah. And then in last kind of cool story that has nothing to do with nudging, but is awesome, there's a bunch of researchers who did a bunch of calculations on black holes and trying to figure out the black hole information paradox. There's like a bunch of papers that just came out, and they've been calculating and these.
I looked at it. I didn't understand it at all.
Yeah. I don't know that I understand it.
They made it sound very exciting.
Yes.
But I did not understand it.
Well, the question is what happens to information when it goes into a black hole? And Hawking and others, they decided to use quantum theory to describe matter in and around black holes, and that gravity would also have to be described using quantum theory. And there's a hybrid approach, like Hawking and his colleagues, they thought they figured it out, but basically they're wrong. They're not completely right. And through not really looking at it as only Einstein's classical or only quantum theory calculations and actually
and Leonard Susskind helped illustrate all this too. Yes.
Yeah, it's basically over time. They found that new gravitational configurations that Hawking didn't include. And they dominate, they get to be more prominent when a black hole gets really old. So black holes age, everything gets older. And as black holes age, apparently they don't want to hold on to information anymore. And it goes from being this quantum system where gravity is treated in a quantum fashion when they're young to it just being gravity and information going in and coming out. And they don't really know exactly how all that happens, but when black holes are really old, they're just like in one ear and out the other. They just do not want to hold on to anything. So they're just like spitting information out all over the place. But this is the new idea. It's this new calculation based on a hybrid combination of physics ideas. But yeah, it doesn't.
I need like an analogy with like a pool table, a rubber mat, and a swinging pendulum. Maybe, I don't know.
Maybe.
Yeah. The worst one. The worst.
I just like thinking
like,
of really old black holes who are just like, I don't hear you, and they spit it out. Whatever.
The worst analogy was the one where it's like, okay, so you have a bathtub full of water, and you put a couple of drops of food coloring in, then you mix it up. How do you get that information out again? I don't. Why would you even try? I don't. Yeah, maybe this is something we should reach out to one of our physicist friends to try to
come and explain this.
Slow-splain me.
Mm-hmm. Yes, Well, maybe we can get Ethan back on. Ethan Siegel is pretty fantastic at that.
Absolutely.
Yeah. And then leading us out of the show, we do have a question for tonight. This is from Fada. Fada says,
Hey, Fada.
Here's my head scratcher ponderer for your TWIS user questions section. This question either shows I'm incredibly brilliant for thinking of something no one else has thought to do, or I'm incredibly stupid and have no idea what I'm talking about or how astronomy actually works. Either way,
That's how I roll.
I think it's a fun thing to ponder. So when our best visual telescopes view phenomena in our galaxy and the universe, be they the Mount Wilson Observatory, the Cerro Paranal or Cerro Tololo observatories of Chile, the Hubble Space Telescope, or the future James Webb Space Telescope, we have to deal with the fact that due to the whole speed of light thing, what we're looking at is a snapshot of how whatever object we're looking at looked that many light years ago that it is distance from us. So when we're looking at like the Pillars of Creation nebula, we're not looking at how it looks now, we're looking at how it looked 7,000 years ago, because it is 7,000 light years from Earth, and it took its light 7,000 years to reach us. When we look at the Andromeda galaxy, we're actually looking at what that galaxy looked like 2.5 million years ago. For all we know, any of these objects could have ceased to exist by now. So the question is, given our best guess as to where the universe's center is moving away and expanding from the Big Bang, and given that we could estimate the general path or arc of our Milky Way galaxy away from that original Big Bang and a universal expansion, would it be possible to get to a point to point a powerful visual telescope like the Hubble back along that path that our galaxy has traversed from the universal center and actually take an ancient picture of our own galaxy from thousands or millions of years ago? Like a galactic selfie? Where's this one gonna fall best?
Wait, we're supposed to answer that question?
Yeah, what do you think, Justin?
Yeah, so I think what we would have to do is travel at the speed of light. Maybe faster. And then go really far away and take a bit. So here's the thing with taking a selfie. We already have a nice selfie of the Milky Way. We should be satisfied with it. We should go ahead and use it as our profile. It's when we shoot towards the middle. We're out in the rurals. It's basically like right there, like Kiki did right there. Yeah, her selfie, her hand isn't in the picture. She takes a picture, right? She's missing her hand. She's missing the entire back side of her. Probably is only, shoulders up, ish. So really, the picture you get, even with a good selfie, isn't that great. And so the just focusing on our Milky Way is basically the best selfie we're ever gonna get of the Milky Way, so our own center.
I feel like you'd have to go too far. Well, I guess if you had an infinitely,
You have to go ahead.
you'd have to have a telescope that's like way better than anything we have, right? Because I think it's all a matter of, if you're talking about light years.
Telescopes are fine. You have to travel. You have to take the telescope many
You have to take the
Right,
telescope
no, that's
to
what I'm saying.
wherever
If
it
you-
is.
I think what he's asking is, if
Can you look back?
were in orbit around Earth with a telescope,
Okay.
could you point it at a direction where you could see the Milky Way before now?
No.
So
No.
even though you're currently
Because you
in
can't.
it,
No, we're in it, so we can't see it before it was here. Because
so
we're
yeah,
in
but
it.
that's the
there's
question,
things-
right?
okay.
Is
Yeah.
that because of the speed of light? Like
Okay,
basically the
I'm
question
starting
of
to
like
catch it. I'm starting to catch it.
when you do a panoramic and you can get yourself in the picture 3 times, right? Because as you move the camera, you run around and go to another spot, right? So it's that question basically. Can you do that with the Milky Way because of the speed of light? But I think it's too far. You'd have to be able, if you had a telescope that was
So-
incredibly sensitive.
No, it would have to
No,
be
no,
faster than the speed of light to go
No.
back to that point in time to be able to take a picture there
And then come back. Yeah. Okay.
and then come
so,
back.
So the idea is, it's sort of like evolution, and we talk about how evolved the thing is. Everything's just as evolved. It's just maybe changed more or less than something else. those things that we are seeing that are very far away, that we're seeing tens of thousands or millions of years and a billion years into the past, is at the same point in time that we are. It's just we can't see it because all we've seen is this light that's taken so long to get here. That doesn't mean that's where it is now. It's just the sort of after image of it that's coming at us. And then that image, after image, is gone. It's not still persistent in space. It's still not sort of stuck there in space. Unless you're even further away,
It's
I guess.
moving, we're moving. Yep.
Yeah.
I also feel like, it's like I don't have any selfies from when I was a baby. My parents did take pictures of me, but that's because they were there then. But I didn't have a camera when I was a baby, and I can't go back to when I was a baby and take a selfie of myself now. That's time travel.
But the answer is also yes. You could, if you could go a little faster than the speed of light and move yourself a billion light years away, and look back at where our solar system or galaxy is.
But I guess
see that you would be seeing that light getting there from our past. But you would have to have gotten to your destination faster than the speed of light because you'd have to beat the light that's traveling now there.
Well, I think it's all about I think the relative distance is the problem, right? Because so basically the question would be like, if it took 10 years to take a picture, okay, and I right now, or I guess it took 10 years for a picture to get back to me. Mm-hmm. And I right now took a picture of Sonoma County, and I could like zoom in, right? And this is a picture from 10 years ago. Am I in that picture? Because I was in Sonoma County 10 years ago, right? That's the question, I think, is like, if you could point a camera from Earth's orbit at the position of where the Milky Way was 1 million light years ago.
We're not there now, though, so we can't capture that light.
Right. And that's the answer. Is that because we're not physically there now? That's really the key. Because we're not physically there now, there's nothing to capture. We would have had to take that picture then, and we just wouldn't get the picture until now.
Yep.
Oh, the other way you could do it is you could invent a time machine that could travel in time but not space, meaning it stays in the same point of space relative to where it was. You can travel 1 million years into the past but not move in space, and then look back and you could see where the Milky Way was 1 million years ago. That's the problem with time travel. I know I've talked about this before. If you go back in time 24 hours, if
different place,
or forward 24 hours, the Earth, the Sun, the solar system's a totally different place. And if you don't have a spaceship to get back, you're just dead in the coldest space. That's why time travel's been invented literally 100 times. It's just every time they've tried it out, they've blinked themselves out into the cold of space and died. That's not the first Tesla that's out there, by the way. Come on.
Yeah.
We know what really happened there.
So what we should do is, what would be awesome is to get a picture now of something here and then beam it out into space somewhere that the Earth is going to be.
Oh, yes.
Like 10,000 years to see if someone can pick up that signal. That would be the way to do it. Transmit a picture to the future from the past.
If you could go twice the speed of light, you'd be going backwards in time, I guess. Because you could get out past I Love Lucy show if you did that for 80 years. And wait for it. And then you could watch movies coming from Earth. Television broadcast from Earth. But you have to go, that's why that's the whole idea of going faster than the speed of light breaks all the rules, because that is actually backwards time travel.
Yeah, because it's warp 10.
The way that we determine where anything is, or where anything is in time and exists, is basically based on the speed of light. If anything moves faster than that, you would be seeing the universe earlier in time.
Just in time. It's the end of the show. Yeah, we made it to the end of our show. Thank you, everyone, for listening. And if you have a moment and you haven't bought a TWIS calendar yet, head over to twis.org and click on the froggy toad and buy a TWIS calendar for 2021. Blair has made arts for you. It's time now for the shout outs. I would love to shout out to people who help support the show. Shout outs to Fada. Shout outs to Identity 4. Shout outs to Gord. Thank you for all the things that you do for the show. And thank you to the Burroughs Welcome Fund and our Patreon sponsors for their generous support as well. Thank you to Donathan Styles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Josiah Zahner, Mike Shoemaker, Sarah Forfar, Donald Mundus, Gerald Sorrell, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Corinne Benton, Sky Luke, Paul Ronovich, Ben Bignell, Kevin Reardon, Noodles, Jack Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Melizond, Johnny Grinley, Flying Out, Richard Porter, Christopher Dreyer, Mark Massaros, Artyom, Greg Briggs, John Atwood, Robert, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavis, Alex Wilson, John Ratnaswamy, Sue Doster, Jason Olds, Dave Neighbor, Coast D. Ranke, Matthew Litwin, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Ben Rothig, Gary S., Ed Dyer, Tony Steele, Ulysses Adkins, Brian Condren, Jason Roberts, and Dave Friedel. Thank you for all of your support on Patreon. And if any of you listening right now are interested in supporting us on Patreon, you can find information at twis.org. On next week's show, we will be speaking with science writer Marta Zaraska about how to grow younger while growing older. Don't miss it.
Otherwise, you'll be the only old person, and the rest of us will just be younger. And
Young. That's right.
But we will be back next Wednesday, 8 p.m. Pacific time, broadcasting live on our YouTube channels, as well as the Facebook channels and whatever other twis.org/live channels and whatever else is out there.
Hey, do you want to listen to us as a podcast? I know you'd rather check out all of our amazing hair today instead. But if you want to listen instead, you can just search for This Week in Science wherever podcasts are found. If you enjoyed the show, you can also get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up for a newsletter.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twisminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS, in that subject line or your email. It'll definitely get spam filtered into the sucker of a suckerfish who's surfing a whale.
Yeah, that's
Where
back.
is actually an opportune place to hit us up on the Twitter. We are at TWIS Science, at Dr. Kiki, at Jackson Fly, at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We will be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
We've come to the end of the hour. Hey, chat room. Oh, I hit the wrong space bar. Then we get more songs, more music, more songs. Hey, chat room. I see you. Hot Rod, I got the email. I did. I'm sorry I didn't email back yet. I did get your email. Yes. Thank you. Thank you. Things look very cool. Yes, yes, yes. How is everybody out there? How's it going? Justin took a break.
Just looking at my phone. Don't worry about it.
Yeah, come. Wait, Vegas? No. Don't tell me Nevada's going. I haven't seen anything.
No,
we can't
nothing's
talk about
changed.
This stuff. Okay, good.
Nothing has changed. Everyone went to bed in Nevada, I think. They're like, we'll deal with it tomorrow. It's fine. They can wait. Like nobody's stressed about it. They can wait. What's one more day between nations?
Wait,
It's fine.
It's fine. It's fine. Yeah. Oh, thank you, Fana. I just don't know if everyone out there. Shubru was asking me earlier if I was wearing my my bathrobe, because earlier it was like after lunchtime and I was still in my bathrobe. I had not yet gotten dressed. I was curled up in my bed for a long time this morning, and then I started working, but I didn't actually get dressed to start working. Just stayed in my pajamas and my bathrobe.
Happens. Yeah.
It was one of those days.
It's flexible. If you don't have any video chats, nobody knows.
I had none. Yeah. It's just it was a mood, and I think there are other people who are feeling the mood out there as well. Right. It all happens later. Slippers until noon. Very nice. I know I'm allowed to be in my bathrobe. And I will have to let you know that this cardigan sweater that I have right now is very bathrobe-like. It's cozy and it has pockets and it's long.
Well, I'm wearing sweatpants, so it's fine.
Yeah.
Can't tell.
Today is the day. My friend gave me this t-shirt.
Oh, yes.
I got a t-shirt from my friend.
That's a good one.
It's a good one.
I thought that's what that t-shirt was.
It is what that t-shirt is. Yes. Slippers all day long. Right now I'm barefooted. Is it barefooted or barefeeted?
Well.
Am I barefeeted?
Bared feet.
Identity 4, I had bunny slippers until my friend's dog ate them.
Oh, no.
Now I don't have bunny slippers. I should get myself some new bunny slippers.
So first my mom and then my dad later were sales reps for the bare feet slippers.
Oh,
Yeah.
the bears.
Yeah, they look like bear paws, and they have
the bear
the
paws.
nails on them and everything, and the bottom of the slipper has the paw print. So they were the sales rep for those for like the 80s and 90s.
What an interesting...
That's funny.
Yeah. So
That's
we had
great.
lots of them, but we don't have any anymore. I'm so sad. We don't have any anymore.
Slippers get old. I mean, you
Yeah.
feet
I
in
just
them and feet sweat.
you don't
and then
know
your
in the
slippers get all
moment.
feet sweaty.
that like this thing that you're doing is going to be like a thing later that could be worth a lot of money. You know, I bet you could sell some of those for pretty cheap.
I think they
for pretty
sell
expensive.
those still at the Black Bear Diner.
Do they?
I've seen bear. Yeah, it's all bear themed.
I feel like they're not as good. These are very like high quality.
You haven't seen them. I love it. I love the team loyalty though. That's fantastic.
Yeah.
Carol Ann, I love this idea. Someone needs to invent a robe that has a camel back with a thermos of coffee with a straw.
Okay,
But it wouldn't be a camelback. It would be like, maybe it would be down like the sides underneath your arms. So it'd be like keep you warm in your sides, and then the straw
Yeah,
would
dual
come up.
purpose.
Like,
Or you can keep it
Oh
warm.
yeah, yeah. So I have seen these at Black Bear,
Wait,
and
what?
yeah, they're not as good.
You've seen the camel back? Oh no, the slippers.
the slippers.
The bear slippers. Sorry, I'm one step behind you all.
Guarav Sharma, thank you. To understand Crocs is a thing that is tougher than that black hole stuff that we were just getting treated. I also don't understand why anybody wears Crocs. Let's create a plastic.
I mean, some
shoe
people
that
really
creates foot humidity to the extent it seems like a bad.
I have a very good friend that wears the like Sherpa lined ones in the winter. They have like the lamb's wool inside of it or whatever.
That's warm.
Yeah. It's funny. And they have the, what are they called? Giblets or something? Or the little
What?
pins? Those are the pins that go into the holes.
Blech. Blech. Nope.
Oh my god.
I'm just gonna say no, not a Rooney. Nope. Good night, Fada. I always end up going outside in my socks because I'm too lazy to put my shoes on. Like, shoes are right next to the door, and I'm like, I'm just go outside. Come back inside, leaves and sticks sticking to the bottom of my feet.
What do you go outside for?
You have to, I don't know, take the garbage out once a week. Then you
I'm
go
happy to
outside
have
pull
the
the bins
dog.
back in. Yeah.
If I didn't have the dog, I feel like I'd never go outside.
Yeah, and so no outside shoes in homes in Scandinavia. I am a no outside shoes in the house kind of person also.
Yeah,
It's much
that's
more
actually
civilized.
it's a thing that I don't think was a thing when I was growing up, but is
No.
a thing in like most households in America, or that I know. Most households
Now.
in California seem to be that way, at least.
for sure. Oh, I see some calendar purchases.
Mm.
There are some calendar purchases.
Nice.
Great.
Nice.
Yeah. So California voting stuff. Is there anything that you are excited about having been passed in California?
I haven't looked at the props today.
So
I heard it
California,
didn't go how maybe it.
for a deep blue state. It's a very not progressive voting pattern in all the ballot measuring stuff.
California's complicated.
California voted down. This is the one that I'm the only one that I'm like the most care about. Like I get it. Like the drivers should be employees. They should get access to benefits and other things like this. And that was, but there was $1 billion spent against it. So I understand that.
Mm-hmm.
Californians voted down the ability for communities in California to vote on rent control.
Yeah.
So they weren't voting to implement rent control. The ballot measure was to allow communities to vote whether or not they would want rent control.
Whoa.
And it got voted down.
What?
And in the midst of what has been a decades-long housing crisis in California, an affordability crisis in California, the people of California voted down their own ability to decide whether or not their community was right or needed rent control.
Is that something that, and is it now like a law on, it's going to be a law on the books that communities
been a law.
can't vote for
No,
it?
it's been a law on the books that they can't. It's been a law since
it was getting rid of that.
80s or something that they can't. It was to get rid of that ridiculous law and make it so that communities could choose to. So they said, no, we don't want the right to decide whether or not it's right for our community. We want to reject the ability for communities to have a voice and a vote over this. Utterly ridiculous.
That's interesting.
It is, and it's come up a few times, and it gets shut down, and it's always very moneyed interests against it. It's always proposed as this is going to lower everybody's housing value, blah, blah, blah. It is the most, whenever you see somebody voting against a thing that very much they themselves need, you're like, somehow you got nudged enough away from your best interest by some sort of nonsense
nudges.
that was intentional. But it's like, it's almost as if it's like, Californians, would you like to vote not to have a vote in your next election? Yeah, I'm tired of having to show up. Just vote for me. Decide for me. I don't want to have a voice. That's what the vote was. It wasn't for rent control anywhere. It was just for the right to vote on it.
But I bet that's
down.
the problem. I bet a lot of people thought they were voting for or against rent control.
Yep.
Yeah, no, because people are low information.
But it's also
the way that
all
it's written
convoluted.
down.
Exactly.
And,
it's like the legalese is designed to be confusing.
Yeah.
It's like, how many words can we use? We're going to use 50 words to describe something that could be explained in 7, so that people
don't
do
forthwith. It shall be allowed.
I do believe that the $5 billion to spend on stem cell research was approved.
That's great.
That's good.
That's fantastic.
Yeah. Eric in Alaska saying, putting on my libertarian hat here, rent control doesn't really work as intended. It tends to add additional distortions into a distorted market. New York is a great example. San Francisco, also a big example of rent control for sure. I agree with that, Eric. But I think that the big point there is voting against your right as a community to vote on the issue. So it's just on the books in California that you can't vote for it at all.
So here's the big argument I have
voting against
for,
your right to vote.
For being able to vote for rent control. is simply if you live in a community where the majority of the people own their homes and might own a second home and rent it, chances are it's not going to pass. If you live in a community where the majority of people in the community do not own the property, but it is a almost, probably 60% or more rental population, meaning the people who own the property probably don't live in that community, that's when that shift needs to take place. That's when you need to be able to vote rent control in to a community that has been unaffordable to everybody living there in terms of purchasing. That's why it's important. And the other thing you have to understand is one of the reasons that the stock market's been booming in the time of an economic hardship is because Wall Street is banking on buying up foreclosures, buying up properties of people who are renting, who had a second property renting and then couldn't rent it anymore above their mortgage because the people they're renting to lost their jobs. They're buying those properties with hard money out of Wall Street investment funds and are going to rent them back to us in perpetuity forever. And it's been a movement that is very visible in California, where corporate and hedge fundy operations are buying up properties and renting them back. So you have communities where you have, I don't know.
It's just ratcheting it up more.
Yeah, that's sort of why our forefathers of our country, wherever they came from, probably left where they left, is because people couldn't afford to own property. It was owned by the king or the state or the entrenched. So anyway, that's my biggest disappointment. Was that one?
Yeah.
Low information, people. So many.
I have to say though, like, you should be able to read through one page of information on a prop and be able to decide if you want to vote for it or not. The fact that you have to read multiple pages to get into the nitty-gritty of what it is, and then read an entire page of the pro, and then an entire page of the con, and then a rebuttal to the con, and then a rebuttal to the pro, and then you have to research who has written the pros and the cons to then decide if they're full of it, or like if they have the same reasoning as you.
Yeah, the ballot paperwork this year was the most ridiculous I'd ever seen. Because I looked at it, and first of all, I had to go grab my other glasses and a magnifying glass because I kind of typed it a little small. But it was just endless. Wait.
Get the magnifying glass.
When you have that many words, I feel like somebody's hiding the lead, and you have to read it all, and it gets
100%.
It was tedious.
Why can't these things be written in like
summarized?
Like,
do you want to be able to vote about a law in your community?
250 words max, and like
plain
language. It could be they're doing it on purpose, but yeah.
And again, I'm also saying this from the perspective of just having come from a nice socialist Scandinavian country where they don't have homeless people.
Yeah.
They still have people who are mentally ill. You see them walking down the street talking to themselves, but it takes a second look because they're well dressed. They're not bringing all of their worldly belongings with them because they have some place to live. It's a very different.
Well, I mean, I think it was Salt Lake. No. St. Louis, it was an SL. Maybe it was St. Louis. I don't remember. But there was a city in America, somewhere between California and New York. I don't recall. That they housed their homeless.
Utah.
That was it was Salt Lake.
Salt Lake City spent the money, and they're like, hey, actually, that was way cheaper than all the stuff that we've been doing.
Not to mention, like, people then were able to get jobs because they had an address. And then they ended up, a lot of them, moving out into the general stream of apartments.
It is such an obstacle.
Help and support. It's huge. It's huge. Mm-hmm. Don't make it an obstacle.
We lack empathy. That's why we got to get the psilocybin
the
in
mushroom,
the water supply.
Yeah. Mm-hmm.
Yeah, psilocybin in the water supply and empathy being taught in the schools. I've seen some really interesting writing though on empathy that I find and I think what was his name? Brian Hare was talking about it a little bit when we talked with him about his book for like 3 hours. But there's been some writing on how empathy is, people have thought for a really long time, it's great because you're able to like see from others' perspectives. But what people recently have been saying is that we're not teaching it in the same way anymore. And instead of teaching people to empathize with others who are not like them and to be able to empathize with people in very different positions and conditions in society, the empathy is there. We have a lot of it. Everybody has it, except it's now being focused on those that are like us. And so we're more empathetic to someone who is like us, who has had something bad happen to them. So for instance, you lost your job or because of, some factory getting shut down or, whatever, or you're empathetic with a family because like your family, they didn't have health care and have massive health care bills. And so our empathy has now started becoming more driving us to greater tribalism because we're not using it to understand others in the way that we can.
This is why I need to get hired someday as a political consultant for putting policy out there, because I can describe that exact same result and desired result from an empathetic perspective, from a completely selfish perspective. Mm-hmm. You can address it through the fear that some people naturally are more likely to respond for. Do you want people who do not look like you, who are desperate for food and money or belongings, living in your community? Or would you rather that they be given the just basic necessities and never bother you? Like if that's what it takes, it's just, talk to them in the language that they can understand.
Or would you rather that everybody has the basic things that they need so that you don't have to worry?
They don't desire yours.
They don't care about your stuff.
Yeah. Yeah, it's not, if that's what it takes, then just go that route. Take it from I completely selfishly don't want poor, homeless, hungry people without health care in my community because I think that the community is healthier and I'm safer if everybody has a place to live.
If everyone's taken care of.
Has a doctor that they can go see when they get that weird cough that they didn't have. But, all of this, yes. Thank you, Daniel Smith. It is too much sense. Why is it hard for people to have the common sense for these things? And part of it is conditioning and that nudging that you're talking about, because there's all these weird things that have been, even when it comes to healthcare, it's like, it's your right to see your doctor, even if your insurance doesn't cover anything when you get there. It's your doctor. It's your right. Like all of this nonsense that gets thrown at people that they grab onto and be like, yeah, I want to have my doctor, even though my insurance won't pay for any procedures done by my doctor. I still want my doctor. It's ridiculous what people get sold on. And I just wish that people all could experience a day or a week of society that is doing things differently. Like America has this weird idea that we're the only ones who've tried anything ever, and we've ruled things out here. We've just ruled them out because they couldn't possibly work, even though it's massively successful somewhere else. And they don't have some of the endemic problems that we have in our society because they addressed them.
Because we could be planning for the future like Scandinavian countries. We could be using our experience with pandemics, with epidemics like Asian countries. We could be, we could be taking experience from all these places.
Well, we don't want to change anything because that's like what we're currently doing is what is fundamentally America, except that we're only like 200 years old. So how can anything we're doing be fundamentally unchangeable when we are such a young country?
Yeah, exactly. And a country that has changed drastically and dramatically many times over.
Yeah,
Who's
we
gone
can't
through
change
it?
anything because then we won't be America anymore. Like, no, incorrect.
Home of the free and the brave people who want to try new things.
Yeah.
Let's do it.
And this is like, we also have to then address things like the criminal justice system.
Yep.
Is it possible
All the
that
things.
America is such a great system, great country, we're doing everything right, and yet we have to incarcerate a greater percentage of our people than anybody else in the world? Does that make sense that we're doing it right? That we're incarcerating more people?
In
Does that
Oregon
make any
now, with
sense?
decriminalization of drugs, hopefully we will be incarcerating fewer people. Not that we have a big population in Oregon to begin with, but it's a step. It's a step.
Yeah, sentences are too long. Support for there is no rehabilitation associated with the prison system in the United States. It is a big business. You're absolutely right, Carol, in the chat room.
Yeah, well,
Eric-
and because it's free labor.
Eric in
It's
Alaska.
slave labor. No,
Yeah.
it's slave labor, and it's
Yeah.
in the Constitution as no one can be forced to work without money except
Yep.
prisoners, which is the only
That was on
form
the
of
California
legal
ballot.
slavery.
What happened to that?
No, the California ballot was going to make more things. Like,
That's passing, isn't it? The allowing ex-felons to be able to vote again?
ex-felons to vote. Yes, that's
Yeah,
passing.
that's
That's a
passing.
good one that's passing.
Yeah.
There's another one. They shot down the eliminating
Is it-
cash bails,
That's not right.
which is
Oh,
I
that
think
whole thing.
the cash bail system is a business that needs to go away. However, the NAACP came out against that because they're like, whoa, you're going to use one of those computer systems that says based on the neighborhood you come from. We've seen that in action before, and it sucked.
Right.
Yeah, so this is one of the
Interesting.
ones where something could have been very simple, but instead was like made extra convoluted and confusing. And the more I read, the more I got confused
And
about
then
what
you
to do
vote
with
no.
that ballot.
Then you go, nope, no, too complicated.
It just, the more I read, the more confused I got, which is not how it should be.
Yeah. There's basic things that we can do as a society that invest in people. Those things like free education. Making education a thing that we do in the national interest alongside the military. Maybe with some of the military's budget even. Is one of those things that we could do that would build a better, brighter, stronger future in this country. The resistance against that never has made a decent argument, in my opinion. I've never heard an argument against educating our population that made, oh yeah, that's why we shouldn't do that. It's never made sense.
Hey, I have a little bit of good news. I mean, this is a biased poll result on Twitter. 358 votes so far for my poll question. If COVID-19 is spiking in your area, how do you feel about lockdowns to control the spread? 75.4% say I'm for it. Only 6.1% say I'm against it.
Mm-hmm.
So, hey, I like that. I like this result. Good job, everybody. But I'm not going to bias these results anymore. So if you have not yet voted, I tweeted out a poll earlier, and you can vote for how you feel about lockdowns.
Again, I'm a very selfish person, honestly. I'm not nearly as empathetic as some people give me credit for. I would like a lockdown just to keep everybody else at home. I feel like that makes my community safer.
Everybody stay home. It does.
Everybody else stay home.
Yeah. I don't know. I'm basically acting as though I've been in lockdown since March. Since March 8th, I think. So I really haven't done much at all. I'm not going anywhere. I think I've been to the grocery store a handful of times. Oh, so everyone, Carol Ann doesn't Twitter. I'm sorry. I Twitter. I tweet. I Twitter tweet. For everybody who's out there, be patient. We still have to have patience for votes to be counted, for science to be done, for processes to take place. Sometimes you just have to wait. And patience is something we should practice and we should get good at. It's good to be patient. Patience is a virtue, is it not? Come on. Let us have virtue.
Well,
Says the person who likes binge watching television shows all in one go. I can't turn it off. I have to watch what happens now, right now.
Oh, wait. This is a thing I have to tune in a week from now to find the next episode? No, I'll wait till it's all on Netflix. Is it that time?
It is that time. I need to go to sleep. I need to get rest. Everybody take care of yourselves. Yeah, right? Do you need to sleep?
I don't remember what that's like.
Go
Say goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin.
Good night,
Kiki.
Kiki.
Good night, everyone. Thank you for watching. Thank you for joining for this live broadcast of the TWIS podcast. We will see you again next week. Have a great week. Patience and sleep. It's good.